soon. The recording will be available on YouTube right after the meeting. So the recording will be uploaded.
Yeah.
Are
you available to join?
Sorry, I can hear someone talking. Okay, so I will now pass the mic to Maxime. Maxime is a professor at the University of Sherbrooke in Canada. He is the president of the International Society for Tractography, and today he will present the Millennium pathways for tractography. Maxime, you have the stage, and we will have a discussion open for around half an hour at the end of the presentation. Thank you very much.
Great. Thanks, François. I want to take the opportunity to thank you, François, personally, and Chiara Maffei for coordinating the Tract-Anat Club. This is great, and I'm happy to see all these new names online and some familiar names. So welcome, everybody. Also, for those watching this through the recording, maybe somewhere in Australia and Asia, welcome. Sometimes we do have Tract-Anat clubs organized on other time zones, such as Asia and Australia. So today, the plan is to give you a brief history of the society, the International Society for Tractography, how it came about from the first tractography and anatomy retreat, the Tract-Anat retreat, which will lead me into telling you a bit about the brain structure and function tractography and neuroanatomy collection, which will be a nice collection of short papers that resulted from this Tract-Anat retreat. And the last paper of this collection is all about the grand challenges of tractography, or what we call now the Millennium pathways for tractography. So the focus of today's talk will mostly be on that last part. But to give you a little context, I'll explain how this all came about. And so I think the original seed really came from the handbook of diffusion MRI tractography. This took Flavio Dell'Acqua and Alex Lehmans and I almost 6-7 years to build. And as a result of this book, we saw that we had a list of 100+ authors for all these chapters. So 900+ pages, hundreds of co-authors. And so we realized, like, wow, there is a community here, and it'd be great to start organizing events, meetings, virtual meetings for this society. So we started thinking about this. And then when Laurent Petit came to Sherbrooke to visit my lab for a year, this is Laurent on the top here, with a postdoc, Graham Little, we really said, let's try to get together and see if we can start this society. And can we create a nonprofit organization, the IST? And so we did. We did this in Canada. This is officially a nonprofit organization. And we got this group of organizers, these 10 people that you see on the page here. A little bit spread across expertise, not only tractography methods developers, but also clinical scientists, neuroanatomists, to organize a program where we could discuss the future of tractography. And so that was the organizing committee of the first Tract-Anat retreat, which brought us all together in Corsica. And we can discuss this later after the talk. So you see the people here, about 55 people came to Corsica about a year ago now. So this is in France, on a French island by the Mediterranean. And we got together there. A lot of Europeans, about 50% Europeans, 30% North Americans, some people from South America, Asia, Australia. And we got together in a week, almost a week of discussions. And let me tell you a little bit more about this. So we had 3 different tracks or themes from neuroanatomy and validation. First theme. Second was all about tractography algorithms and methods. And third was about clinical applications and connectomics and clinical translation. And so we had experts across all these 3 fields. And really the goal of that retreat was to focus on what can we achieve only by working together. what can we achieve just beyond my personal lab or your personal lab, but as a community? What are the challenges? What do we do well? What do we do less well? And what are the next short-term goals, mid-term goals, and long-term goals for the community? And just to give you an idea, this is what the program looked like. So very little formal presentations, no poster session. It was all about roundtables, discussions, and getting together. And as a result, and you can see here, we played some fun games, and everybody participated and prepared slides about something the did you know game or the misconception game. So everybody in the crowd presented something that they thought maybe was interesting for the rest of the crowd. And so that resulted in, and as you'll see later, as 21 did you know short papers. Then we had some debates about questions, and that led to morning sessions of debates in roundtables discussing pros and cons, local modeling, DTI versus crossing fibers, tractography algorithms, different validation strategy, different application. And so as you will see, these debates now are leading to 9 new short papers in our brain structure collection. And finally, we had roundtables about the Millennium pathways for tractography, the grand challenges for the future. And that's what I want to focus on for the rest of the talk. And so a year later, we are almost there. So thanks to Kurt Schilling, Laurent Petit, myself, and a lot of people involved, we now have something called a collection at Brain Structure and Function. So we have a collection created called Neuroanatomy and Tractography. And under that collection, very soon, within the next few months, you will start seeing some short papers coming out that are basically the result of our discussions at the retreat. And so we have more or less 9 debates papers coming, and the debates committee was led by Kurt Schilling and Chiara Lefay. We have 21 did you knows. So these are fun facts or known facts that are success stories or open challenges in the world of tractography. So that's 21 short did you know papers from neuroanatomy to methods to applications. And so we have a very dynamic committee there leading this effort by Joseph Yang, Tim Derby, Jessica Dubois, Vinod Kumar, Donald Tournier. And finally, Derek Jones, Kurt Schilling, Laurent Petit, and myself are putting together the Millennium pathways for tractography. And so that's today's presentation. So just to give you a little bit more, so at the retreat, we were getting together. You see the pictures here in small tables of 8, 9 experts. We had 6 roundtables. So we had 3 sessions. And so if you do the math, that's 6 times 3 sessions, 18 roundtables times 3 topics. So in the end, we got 54 recorded challenges that we all discussed together and put in a Google form. And then Kurt Schilling, Laurent, Derek, and myself summarized them. There was some sort of overlap, and we boiled this down to 40 grand challenges, or what we call now 40 Millennium, an analogy to the Millennium mathematical problems, so the Millennium pathways for tractography. So this is extremely hard to present. I presented them for the first time in Kyoto at the diffusion workshop a few weeks ago. So I'm going to animate them, but this is recorded, so you can always go back. This will also be published in a paper. But essentially, that's a roadmap that we can go back to as a society, as a community, to try to see in 2, 5, 10 years if some of them and some of these pathway challenges have been solved or have been partially solved. So at the middle, we have our Tractography Millennium pathways, and they're spread across the 3 topics, the 3 themes. neuroanatomy, tractography, and application. And we have 7 groups that have been organized to organize the 40 problems together. First, under neuroanatomy, we have a group on connectivity, characterization, and validation. And I'm going to zoom into each one after. Then, merging anatomy and tractography, we have linking the neuroanatomy to tractography. We have a box on standardization, accessibility, dissemination, all about communicating better across tractography and applications, how to train the next generation of clinicians and medical doctors. Between neuroanatomy and application, we have a box on normative modeling across the lifespan. How does the white matter change across the lifespan? And what are the open challenges there? What are the challenges in clinical translation of tractography methods and validation methods? What are the informatics side of things? How can we leverage the latest technology and data across scales and species? And finally, a box for methods development, which has a lot of short-term, mid-term, and long-term. So the color code here, sorry for the colorblind people, is green is short-term goals, so probably lighter for the colorblind. Orange is mid-term, and red is longer term. This is, of course, a bit hard to classify. And as you're listening, if you do have comments as I'm going through them, please email us. Don't hesitate. You can join the group, you can join the paper, and we can adjust these things. This is still not fully published yet and still work in progress. So the first block, connectivity, characterization, and validation. So what is the methodology to validate and quantify connectivity at different scales? What is the scale, or what is the multi-scale approach to describe a graph in the brain? And what is the number we put in them? How do we build grand truth phantoms and virtual brains to validate tractography methods? How do we factor in the functional role of white matter? We're pretty good at knowing which part of the cortex is doing what. And here you're seeing an image of the famous motor homunculus. But even in that sphere, there was a latest paper in Nature 2023, which has reinvented a little bit the motor homunculus. It's a lot more of a continuum now that it's taught on the motor homunculus on the cortex side. But we have big question marks under what's the underlying white matter there? What are the functional roles of white matter connections? And the grand challenge is also to include all the multimodality. What can we get from MRI, quantitative MRI, that we can feed into connectivity? I love this figure here because we see a bit of everything. We see the cortex, we see short association tracts, we see maybe connection through the gray matter, we see different layers of iron deposition, we see the vasculature, different cell types. And so this is a bit of a summary of the grand challenge to characterize intracortical connectivity. Next, linking anatomy and tractography. Like this picture here showing really the evolution of different images across species, across scales, all the way from a mouse brain to a macaque brain to a human brain. How do we clarify pathways of tractography? Establish histological and anatomical references for us. Improve tractography in areas that are changing dynamically, either with development or with aging. And how do we develop atlases that really reflect variability across populations? So lots of challenges here linking anatomy and tractography. A lot of the challenges at the retreat were around communication, talking to each other, better communicating, better educating ourselves. And this is in a sense why we have the Tract-Anat club now. We want to learn more about anatomy, more about application, more about methods. And so how do we disseminate tractography better? Here, this image is just a map of the Tractography challenge in the Nature Communication, where you see groups that actually participated. So it's just an illustration to show you that a large part of the world is not publishing or using Tractography at all. So how do we increase this coverage across the world, but also across disciplines, the underrepresented, the low and medium income countries? How do we bring in industry? The industry is getting more and more involved with Tractography. There are products out there getting onto the market. This is an example here of an MR-Trix Tractography pipeline implemented in Medtronics and an automatic track segmentation reconstruction to show the most important tracks around a brain tumor or glioblastoma. And so how do we get our methods into the industry? And how does the industry also communicate what the hospitals and clinicians want? So we want this communication vice versa. And most importantly, how do we get the regulatory approvals for these tools? Building software for medicine is like building drugs. We need FDA approval, or in Europe, different types of regulatory approval. And so how do we get involved with the FDA? How do we get products FDA approved that can propose reports of white matter? And this is an example from IMECA. But to my knowledge, there are 2 or 3 commercial products now out there able to sell DTI white matter Tractography products to the clinicians. And so, and in the end, what are the standard operating procedures to teach Tractography and bring Tractography to the clinic? And so this is a AI generated image, but you can see the point here that eventually we would have guidelines and SOPs to teach resident medical students and have the proper tools to use quantitative Tractography reports in the clinic. The next box is about normative modeling across the lifespan. And so can we have a normative reference of the evolution of white matter across age? So for instance here, the motor tract or the pyramidal tract, how does it evolve from age 20 to age 90 in terms of mean diffusivity or fractional anisotropy or all our advanced multi-compartment models? Can we have a public reference, a little bit like the doctors have a reference for kids' development with head circumference or size that quantify how should a normal white matter brain evolve with time? And then how do we leverage this with sophisticated method to do proper normative modeling and personalized medicine? So medicines for N equals 1. And what you see here in this graph is kind of a mock-up from a brain normative modeling paper, and I photoshopped a bit of fiber tracks in there to see if we can follow and monitor evolution of quantitative measurements of white matter in time, and if we have an accident, a traumatic brain injury, a stroke, see how we deviate from our curve and how we are able to recover to our curve. So lots of development needed here, which would greatly help clinical translation. And so the box about clinical translation is all about bringing Tractography into the clinic. It's already starting to appear in some applications, mostly in surgical planning, deep brain stimulation, neurosurgery. But we can see now it's starting to appear also in traumatic brain injury, stroke. And so the challenges here are improve the methods, yes, but also have a discussion with clinical translation. Formulate an expert advisory group for clinical translation of Tractography. Develop Tractography guided biomarkers for specific clinical trials and specific drugs. And in the end, have better tools that deal with pathology. How do we deal with white matter lesions, multiple sclerosis lesions, holes in our mask, tumors, atrophy, swelling? Our methods needs to be improved to be robust to pathology if we want to bring this in clinical translation, because in the end, it would be great, and that's one of the great challenges, is to have tractography part of the medical education for neuroradiologists and neurologists, where they no longer just learn anatomy from 2D slices and cortex, but they also have white matter and connectivity information to learn medicine and bring this into their clinical practice. Two more. One about informatics. How do we leverage the latest technology? Here's I think a nice figure that illustrates GPUs, AI, acceleration, parallelism, maybe quantum computing. How do we leverage the latest state-of-the-art technology that are doing so well in other applications, especially in imaging science or data science, into the tractography world? How do we leverage these techniques to improve tractography, not only with machine learning, but everything else we can do? And how do we propose new methods that can work across scales? How do we go from really the micron scale to the mesoscale to the MRI scale and have references for this? So a little bit like the Allen Brain Institute, something similar, but for tractography, where we can have light sheet microscopy of individual axons to optical coherence tomography or PLI of mesoscopic scale axon reconstructions, and then macro scale diffusion MRI, and talk about these scales, have methods that communicate and that leverage information learned at smaller scales for higher scales to really improve the a priori knowledge that we feed into our tractography algorithm. And can we build a system, also a benchmarking system, that's incorporated in the life cycle of development of tractography methods? That way, we have high quality tractography methods that are constantly evaluated and benchmarked to make sure that as our image acquisitions improve, as we change parameters in our algorithms, we still have the very high quality results that is expected with great test, retest, reproducibility, but also small variability that can tease apart differences in white matter microstructure. And finally, methods development. This is, of course, a little bit closer to my heart, as I have an identity in this world of creating better methods, improved methods. Tractography in the full brain, in the brainstem, in the cerebellum, in other organs, kidney, heart, breast. There are challenges there. How do we really disentangle fanning from branching, from crossing, from dispersion, from touching? This is still an ongoing challenge for local tractography methods, or even global tractography methods. And in the later run, can we build a smarter kind of GPS system? How can we inform our tractography with all the multimodality information that we have, the priors that we have from anatomy, the priors we have from functional imaging, and can we start including information flow and synchronization across the networks? Include multimodality, like I said, microstructure, and to really have an adaptive, intelligent tractography that does it all. So this is very ambitious, and I love this picture that's in Alessandro Daducci 2016 paper, where he represents the brain a bit like a big plumbing system under our cities, where we do have the tractography, the structural map of all the pipes, but then the size of the pipes is the microstructure, the function of the pipes at the end of the pipes is more about the multimodality and fMRI. We could even include the vasculature in there, the metabolism from PET, because in the end, we want a multi-scale, multimodality tool for the future of connectomics and chronectomics, right? Connectivity in time. And so this is very ambitious, but how do we bring, the real GPS back into the tractography problem? So the future looks bright. We've come a long way. It's about now 30 years of tractography development and a lot more to come. And this is why I hope I've convinced you that we created the International Society for Tractography, really to connect people together, to shape the future, to use this virtual platform to discuss together, but also to organize educational events, groups that start working together to really shape the future beyond our individual labs. And so I want to thank our scientific committee. This is the first version of our scientific committee where, as you can see, we are a community or a society doing more than organizing a conference every year. So we're already starting to work together with these leaders on the educational part, the events part, yes, the technology part. Ariel is leading a standardization group. Lauren O'Donnell is leading an interdisciplinary definition group about the definition of different white matter tracks. Catherine LaBelle is leading an application and translation group. And if you go to the website, you can see all the people involved there. And Alessandro Daducci is leading a validation group. We have a nice set of advisors from, around the different topics, clinical anatomy, surgery, clinical scientists, industry users. So thanks everyone. Derek, Pratik, Kathy, Silvio, Shauna. And so we don't even have a year yet under our belt, but we're our group, we're shaping up, we're organizing ourselves. We're going to be communicating with the brain structure and function collection. And keep an eye out for the call for abstracts. This is coming out very short. You can already save the date. We will have our first in-person IST conference in Bordeaux in 2025 in October. So that's in a few months, 6 months almost. So there will be a call for abstracts for students to present work. And we will open registration in the next month or two. We have an organizing committee meeting and actively working on this. So keep an eye out for that. And with that, I'll break here and hopefully we have a nice discussion or question period on the society and on the Millennium pathways for tractography. Thank you.
Thank you very much, Maxime, for this great overview of everything in the Millennium Pathway Challenge. I hope everybody appreciated it. A very popular talk, so 90 people are here, so that's one of the popular talk we had. That's great news. If you have any question, you can write them in the chat, and we can read them, or I can tell them to Maxime. If you want to raise your hand and wait your turn, you can unmute yourself and ask a question. I would like to open with one question that would be about the clinical translation. I assume that with your experience with Imeka, the company, and as a researcher, it seems like the way in for some of those clinical translation is a killer application, just like for DWI with stroke. Is there one for tractography, or do you think we should aim for an application like that, like one pathology or one use case that we should put our effort on to kind of sneak into like the clinical world for tractography?
Good question. That's something we talked about in Corsica. The quote unquote low-hanging fruit is clearly neurosurgery. Neurosurgeons are using more and more tractography to plan their surgery. And there are two worlds there. Really, for tumor resection is one thing, but the whole epilepsy world and deep brain stimulation, where surgeons are putting in electrodes, seems like a powerful first way for tractography to show successes. Having said this, it's quite a small niche as well, right? Neurosurgery is a small niche. in the whole spectrum of medical application. But this is, I think we know that this is where tractography has a lot of promises and is doing well. I have a feeling that the neuronavigation companies are already on the case. They're working with us, they're working with neurosurgeons, they're working with the MR-Trix group. So this is great. I would love to see a bit more. I have a feeling, and maybe I'm wrong, but that there's something to be done with normative modeling about even having a good quantitative DTI report that quantifies the normality of certain white matter areas in the brain that can be used to detect anomalies and monitor them in time. I see this as a nice killer application for both pharmaceutical companies developing drugs and also for clinical practice. It's just that we have to make our way in, and that's difficult. Making our way in is showing successes through our science, communicating it better, convincing clinicians that they need it and that it's useful. And the big challenge there is that people are not necessarily going into MRI, right? There's only a few pathologies where people are actually going into an MRI. Otherwise, they're not going into the MRI machine. So that's also a big roadblock for the killer application. We need an application where patients go into the MRI.
Thank you for this great answer. I see someone raise their hand, so I will. You can go ahead, unmute yourself, and you can ask directly, Maxime.
Thank you. Hello, hi, Maxime. It's Francesco Vergani from London. Yes. As a neurosurgeon, I was very happy about the comment you just made about neurosurgery being one of the fields where there can be perhaps a more immediate translation of the advantage of tractography. And I guess that as neurosurgeons taking part in this community, I think there are two things. The first would be to work on validation and standardization, which I think is still quite lacking in neurosurgery, because you can see across different centers, people do tractography, but it's not necessarily a very, or let's say, a good level of tractography. So I think that there is a little bit of teaching there that neurosurgeons may still require, and therefore it is useful to have them on board within the society. And the other thing is to develop methods and see what really tractography can do, apart from the obvious planning of surgery and study relationship of white matter tracts compared to tumors or where you want to do resective surgery. And this is, again, perhaps the most exciting. So I just wanted to make this comment, but it's great that we are working together, I guess. Thank you for everything that you're doing.
And thank you, Francesco, for your involvement. So just for everybody knows, Francesco right now is leading a survey with neurosurgeon colleagues across the world to try to evaluate what is everybody doing in their center. And with Silvio, I think, and Joseph Yang, you guys are organizing maybe a paper to discuss this. And then, this is an important thing for the society, is to coordinate ourselves. And like I tried to say, hopefully one day we have guidelines. Guidelines in terms of methods, but also guidelines in terms of usage.
Or a minimum requirement, minimum standard of things needed. Thank you.
Great. Also, I'm not following the chat. Sorry. Do you want to monitor?
Yeah, so there was a few comments in the chat. So someone mentioned that they are a neurosurgeon and that there's a lot of industry work going toward a tractography solution, but it's often coming with like a black box approach. So like the company is providing tools, but you cannot really play likely with parameter, or you cannot change something depending on your personal preference. So it would be great to develop a standard and like, again, open source tool rather than like a black box from a company. So I don't know if that person wants to contribute.
Thanks for the comment. Yeah, don't hesitate to open your camera and show your face. But one thing to point out, and we do have a chapter on this in the handbook of tractography, is as soon as you have pathology, most of our tractography default tools start breaking apart. The parameters need to be adapted, the seeding regions, the masking, and so this is really, really challenging. And that's what I meant also about leveraging what's happening with all the new technologies. But as we improve AI segmentation of these different tissue classes, for instance, this will greatly help tractography, because tractography will be able to use these segmentations of edema, necrosis, infiltration to really tune the algorithm and adapt smartly to the different pathologies. So it's a challenge, because keep in mind that most of our methods are developed on young, healthy brains and tested, and I'll be exaggerating here, but on the Human Connectome Project data. So we need to be better at this. We need to confront our tools with crappy data and clinical data if we really want our methods to translate.
There was a question in the chat. I'm going to reformulate a bit. Do you have any strong opinion on harmonization at the scanner level, for example, versus post-processing harmonization? Like, do you believe that the ISP should actively work toward trying to help the scanner constructor to provide tools and more transparent processing so that tractography can be more reproducible straight from the scanner instead of being required in terms of post-processing or even combat when you report metric, for example?
Yeah, that's a tough one. That's really a tough one. This comes back to my, the ISMRM. So the International Society for Magnetic Resonance in Medicine has a diffusion study group. I was president, I think, 2015, and we started this effort of building recommendation and best practices for diffusion acquisition. And these papers are still being written at the moment, but we did survey the whole community, the industry. I've been lobbying and meeting GE, Philips, and Siemens to try to have default sequences at the console that someone could just drag and drop that are suited for quick DTI or a bit longer, more high res, hardy or multi-shell. And this is not easy to do. But I think the timing is right. The vendors are open to this for the first time. And so, for instance, at Philips, now they have in the next release, they will have 3 different sequences thanks to recommendation from this consensus paper. So from work from Carlo Pierpaoli, Shauna, Kurt Schilling. And so if then Siemens follows and then G follows, this would already be a first step. You're going to tell me, yeah, that they have different reconstruction algorithm, different parallel imaging. That's all true. But if at least they start with the same spatial resolution, TETR-ish, the sum number of direction, the same p-values, that's already a layer of harmonization less that we have to do in terms of post-processing. Maybe I'm a bit pessimistic, but I have a feeling we'll need a little bit of both in the future. So both better harmonization at the console, at the MRI level, but also I see in my research or in my company, the harmonization is critical. If we're starting to pool data or having normative reports of white matter, a small amount of harmonization is essential if I want to compare. Even myself, traveling around Canada, my fractional anisotropy in my corpus callosum is changing across Canada, even if we try to harmonize the sequence. So I believe some level of post-processing will be required for a few years still. Yeah.
Thank you for the answer. I see a lot of people in the chat, in the meeting list that are developer of cutting edge methods. I see Alessandro with Commit. I see Gab Girard with particle filtering, tractography. There's a lot of people that develop really cutting edge methods. Do you think it's realistic to think that, you know, at some point, if we come up with a consensus that like the best way of doing tractography is probably very complex, encompassing method like Commit, for example, like, would it be realistic to expect at some point that this would be available in the clinic, or should we, like when you talked earlier about the low-hanging fruit, do you think this is just aiming too high, for example?
No, I think we should aim high. But I also think we need to go beyond our good papers and our good GitHubs. Personally, I see that, yeah, collaborations with clinicians are great. This helps make nice papers. This helps your clinician, but it doesn't turn this into a clinical tool. If we are serious, we will need startups, we will need companies, we will need products that make it to market, because otherwise we will still be doing it for our surgeons, doing it for our collaborators. So yes, cutting edge methods will make it. I really believe so. But we need a transportation that turns these things into products. And I'm not a fan of adopting a single technology or single method. So with, I don't know if Yele Verart is online or Franco Pistilli, or Ariel, but we hope that one day we have a great benchmarking system, right? A little bit like our initial idea of the tractometer. So no matter what method you propose, well, you quantify it, you evaluate it on the benchmark system, and you convince yourself that you're as good as the reference, and you're good. You don't have to use my tracking method. Your tracking method might be as good or a bit better, but you need to benchmark it or quantify its quality. And if you do so, then you can start translating it to the clinic. So that's what I would love to see in the life cycle of methods development, is better evaluation and validation of tools.
Thank you very much. There is a raised hand. Etienne, if you want to go ahead.
Yes, thank you. Yep. Similar to the one of Francois, about mostly open science and open data for tractography, more specific. So I think nowadays we have a HGP dataset that is quite available to most researcher, and for some researcher, there's also UK Biobank, which is not completely open. But I see sometimes it's, and we have more open atlas. So for example, O'Donnell Atlas, Atlas for tractography. So I think this is really helpful. But sometimes I think some of the microscopy or validation data, for example, I think about big brain or those type of acquisition, are not always open science and maybe hard to reach to those people. Do you see a way forward to maybe have better collaboration, or find a way to have this crowd also bringing some open data and have more way to validate or compare result?
Yes, I do. And this is a vision of our technology tech unit or tech group in the society. This right now is led by Graham, but Joël Lefebvre is involved doing a lot of microscopy work and OCT and all the reconstruction algorithms, the processing, the data. One day, or right now, is open. If we had the resources, we would aim to do something a little bit like the Allen Brain Institute, where we would, across scales, and this includes also clinical data, to try to start building a repository of open access data. Minimally through the IST, we should link and communicate what's available out there. I think there's more than you think it's available out there. For instance, I didn't even know there was an open science tumor database with diffusion, with pre and post data that I started collaborating with Joseph Yang. And that's how we started working together, because we didn't have the ethics to share our own patient data. And so as we have more of such open neuro or open science data, either on the macro scale, clinical scale, or meso scale, or micro scale, that's the hope. So the Big Mac data from Carla Miller and Amy could be one resource with PLI, and the databases with OCT. People in Bordeaux will share their MRI data, their light sheet data, their microscopy data. So we're starting to organize ourselves. But right now, the IST is all community driven. It's, it's pro bono. It's volunteer work from all of us together. So at the moment, we have no funding to hire staff or to build data servers. So it's a challenge. We kind of have to do this as a community for now.
Thank you.
Thank you, Maxime. I see Kurt added a question in the chat. I don't know, every meeting so far, we ask Kurt to ask his question, but I can read it instead, unless Kurt unmute himself.
Sure. So I'm sorry, I asked like 5 or 6 questions there, but what struck me was you mentioned this is not easy to do when you were talking about vendors getting this, the recommended, even just the DTI protocol. So I asked a few questions. What was the biggest hurdle? Is it that we haven't convinced them of a specific application? Are they already happy with their diffusion sequences? The competitors don't have this, so they don't have to. Yeah. And then would this be more challenging for a standard tractography protocol if that were to exist, or whatever that looks like?
Okay, so in my discussion with vendors, the biggest thing is that all the vendors said to me, I don't want to dumb down my sequence for you. Like, you're asking me to dumb down what I do better than others and vice versa. So essentially, and yeah, finding a consensus or a common ground is, for all of them, is kind of dumbing down what they can do better. So they don't like it. And that was their first response 10 years ago, to like, go away. But then there was some pressure on them, because in my company, we were running clinical trials, and based on ANNI open data and PPMI open data, I was showing Philips that their Philips system was always off the charts compared to Siemens and GE. And I was telling them, now, look, guys, we're going to not include Philips sites in our clinical trial. And so suddenly they're like, oh, whoa, okay, let's talk now. And that's too bad in a way. I had to pressure them, and I had to kind of, give them the baton or the... But that certainly started to shake things around. And then the Human Connectome project arrived, and the Magnus system in G, and suddenly now they have a feeling, I don't know why. It's probably also powered by open science and reproducible science and transparent science. Now they're listening a lot more, and they agree, they will agree, I think, to have a dumbed down version of our sequence that is common across most sites, which will help in the end clinical trials, will help drug development, will help the industry, and will help sell scanners in the end, more scanners. So it's a bit biased because it's my personal view on this, but I think that's the biggest hurdle. They don't have the commercial incentive to do this. The commercial incentive is to have the latest cutting edge stuff that is better than other vendors. But as a community, I think we need to convince that we need to do just good DTI across the world. Because in the clinic, they're still doing diffusion weighted imaging with pseudo ADC in 3 directions. But if we have a decent, fast DTI sequence that everybody can do and that's reproducible and transparent, I'm optimistic that we can achieve this, and then work from there. Then go to the more advanced multi-shell or B tensors in the future.
Yeah, I wonder if we could take inspiration from the spinal cord diffusion community. So Julian Cohen-Haddad in Montreal, where he had this base sequence, but then he said, oh, if you have the ability to do reduced field of view, if you have this set of coils, if you have this, here are all the options to do these advances, but here's what you don't want to do that kind of set the minimum recommended protocol.
That's a good approach. Yeah. And that's a bit, Kurt, I know that's a bit how we're trying to write this thing up, right? But yeah, it's slowly but surely. But that's why we need a community effort. That's not going to be done by a single person.
Somebody in the chat, great suggestion idea is that where can we find those diffusion protocol? At their hospital, they are using the ANI diffusion protocol, and it's available on their website. They put a link in the chat where when you click on it, you can see that there is a few files that seems to be PDF, probably with instruction, but there is one for NE4, NE3, and one column for GE, Philips, and Siemens. Is that something that would make sense on the IST website or in the form of a spin-off, not necessarily a spin-off website, but something related to the IST to say, like, these are the instructions if you want to follow the IST recommendation?
Well, yeah, I don't want to step on the toes of the ISMRM efforts and the diffusion study group effort, but so Rembrandt, this is an effort from the diffusion study group that will come out in the near future. Kurt, I know you're helping on this one with Carlo Pierpaoli and Shauna. I don't know if they're online. They could give us an update, but I'm trying to push Rembrandt to have such a thing coming out with the paper. I'm also trying to push the vendors to include exam cards or executable from Siemens that someone could download and just drag and drop into the console. And I think, again, that's something Julien Cohen-Haddad is doing well with the spinal cord. They're providing PDFs, the details of the sequence, but they also have pre-compiled sequences that one can just download and drag and drop on their system. So I would love that. That's a great suggestion. And unfortunately, at the moment, these recommended protocols are not published yet, Rembrandt. But you can reach out to me and I could put you in the loop with other people.
And you said this would be more on the ISMRM side because we can probably do tractography with the recommended ISMRM protocol.
And I mean, we're not in competition here. We will find a home in our website to, link it and maybe cross-reference it. But yes, this can help everyone. And these recommended protocols are not necessarily, made for tractography, but as you say, Francois, they could very well work and be.
That's great. I don't see new question in the chat. Is there anyone that want to pitch in? I'm looking at the name, trying to ask someone on the spot. I don't like doing that, but if there is someone that want to ask a question, feel free to just unmute yourself and go ahead.
Olivier just popped up. Salut, Olivier.
Hello. Salut.
Well, we have 2 minutes left, Francois, but yeah, one last question if you want.
Well, we didn't hear a lot from people doing microscopy. I see Joel in the chat. I see Caitlin Rushland. I don't know if I know Maxime and I have our algorithmical computer science bias. Is there a question related more to microscopy or tracer? If somebody would like to push.
May I have a comment?
Of course. Yes, go ahead.
Okay, first of all, thanks, Maxime, for the recap of all the initiative and the background about IST. I would just do a comment about one of the line you sketch for the future about the normative modeling. I believe that we have even a more, let's say, exciting perspective given all the development of AI about building kind of a foundation model, not just a univariate normative model. And so I believe that that one should be considered another very promising, let's say, trend to pursue with the tractography.
I agree, Paolo. And this is more AI flavored, but yes, I agree. And we could definitely add this comment. So just so you know, each line of these challenges will come with a paragraph that Kurt, Derek, and I and Laurent are drafting. And so we could definitely include either a new line or a clarification that these foundation models, yes, are promising for normative modeling. And this is a bit also connecting with the informatics side of leveraging the latest technologies out there for all the other challenges as well. So thanks for your comment. And if you agree, I might reach out to you if you want to contribute to this box and to this comment. Thank you, Paolo.
Well, I don't see any new question. No one seems to be raising their hand, so maybe that's a good point, good time to finish the meeting. So thank you again to Maxime for his great talk and for his presentation about the IST as an organization, but also the Millennium Challenge for tractography. Next month, we'll have a meeting that will be in the form of a panel, if everything goes well, about the new algorithmical development in tractography that makes it more accessible to the clinic or more accessible to researcher. So it's all about accessibility of tractography and to break the myth of tractography being a very complex and processing intensive modality. So thank you, everyone. And if you have any comment about the invitation, the mailing list, you can send me a message.